that is that on prokaryotes and eukaryotes, just trying to make a clear distinction between the two. So our focus is actually now going to shift to eukaryotic cell, but this time around I call it, let's take a tour of eukaryotic cell. Let's try to look at the different parts of that machinery called cells and how it operates and performs its functions. I said every cells have many smaller structures inside of them called organelles. Organelles means little organs, just like humans now. Inside of us, we have various organs that works towards forming system and ensuring that functions are being performed and we stay alive. In a cell, we have organs as well, but these organs, we call them little organs, and that means organelles. So every cells have many smaller structures called organelles or little organs. But the thing is this, depending on the organelles that are present in a cell, that is exactly what determines the sort of function that that cell can perform. Take for example, a cell that is very rich in mitochondrion, that has a large number of mitochondrion, that is going to be an active cells where ATP or energy formation is prioritized, simply because the cells are constantly working there, so they are of course in need of much energy. But in cells that are not always active, of course you can expect lesser number of mitochondrion. Okay, let's talk about let's take cells that secrete, that releases chemicals, as we call them glandular cells. When I see a cell that is very rich in Golgi apparatus or Golgi body, I know that this time around chemicals or substances have been manufactured in the cells and needs to be exported out of the cell. In this case, when a cell is very rich in Golgi bodies, I know it is a glandular or a secretory cell. So the thing is this, the function that a cell will perform is dependent on the kind of organelle that is present in that cell. But that cell, you see, I said not every cells contain every kind of organelle. The functions of the cell depend upon the number and the kinds of organelles present. So what we're now going to do is we are going to take diagrams for animal cells and plant cells and look at the various little organs, that's the organelles they contain, and then subsequently we'll talk about a variety of cells. But let's draw. As a biology student, you must draw. You must draw because that is what retains, those images, those pictures. There is no cutting corners therein. You must just draw. So come along with me. Let me show you my drawing skill. Okay, trying to draw animal cells now. I just go like, okay. Okay, now I have this, and then I have this large nucleus. Okay, let me make this my nucleus, and then I have my nucleolus therein, and then let this be my chromatin. I got this drawing from one of the very best biology textbook that we have in the country, that's talking about college biology by Idodo Ume. If you don't have that biology textbook, you're actually, I think you're slipping on something in biology. It's a must. You, as a biology student, it's a textbook you must have. When you have this textbook, you begin to study biology not from the standpoint of trying to cram or memorize, large chunks of information. You begin to study biology from a systematic standpoint where you understand the concept and how they link. In fact, all of the terms in biology suddenly become synchronized, and you can actually flow. You begin to study biology from a standpoint of wanting to understand, wanting to know. Also, but I have other textbooks too. I have Functional Approach by MBV Robert. I also have Eat Biology by Rotundo. Quite a number, just to mention a few. I have here with me Raven Johnson. I have Raven Johnson, but I have quite a number of biology textbooks which I've studied over the years. What I'm saying is that in Nigeria currently, this is our best biology textbook. It is college biology. I've read it inside out and I know college biology by Idodo Ume. Let me see. I think I have a copy here. Yes, I have a copy here on my table. College biology by Idodo Ume is one of the best biology textbook that can help you to ace your next exam. Very, very important biology textbook. I read it while I was in secondary school, and I know how it has shaped and helped me biologically. So let's continue with the drawing. Okay, this is going to be our nucleus. The nucleus, links up with the endoplasmic reticulum. There are no two ways about it. In biology, you must draw. Or if you're not there, let's say you must sketch. You must sketch. Talking about the cristae of the mitochondrion. Okay. So here is the drawing. This is my cell membrane that surrounds the entire cytoplasm. This here is my Golgi apparatus. This here talks about my centriole. Two bodies at 90 degrees to each other, usually perpendicular, that helps during cell division. This is my centriole. This here is my nuclear membrane. This here, it talks about my chromatin. And this here is the nucleolus. This here is the nucleolus. These three combined, that's the nucleus. That's the nucleus. This here is my rough endoplasmic reticulum. This here is my lysosome, that bag that contains the hydrolytic enzymes in the cell. This here is my smooth endoplasmic reticulum. This here is the powerhouse of the cell, talking about the mitochondrion. Okay, this here is the ribosome. And of course, let me look for that space. Okay, this here is my semi-fluid cytoplasm. Okay, talking about a cell, this at least to on a normal level, at the basic level, this is how best we can represent the structure of the animal cell. Okay, animal cell. You can see the cell membrane surrounds the entire cytoplasm. You have the nucleus seated comfy at the center, and of course you have, you know, numerous other organelles. You, as a biology student, you just must practice to sketch your drawings, as that is what retains, you know, vividly what some of these structures look like. You cannot be a real biology student for real, and not be at least able to sketch to show the main features that show the main organelles of a cell. For example, here we have the skeletal framework of the cell. Like, you ask yourself, what is holding this into space? Why is this not falling down? What is holding this at the center? Why is it not falling down? What is holding this? Why is it not falling down? That talks about the cytoskeletal that talks about the cytoskeletal framework of the cells, which in the course of explaining or taking a tour, you know, moving around the cell and trying to describe the various organelles, I'll talk more about the cytoskeletal framework of the cell. So this here is a proper animal cell. So in case you're wondering, I have so many textbooks I use actually. I have my Raven Johnson. I have Raven Johnson. This here is my Raven Johnson. Let me show you. This here is my Raven Johnson, quite old. I have my It's Biology by Rotundo this year, and McLaurin. This year is my It's Biology. You can see this here is my It's Biology, and quite an interesting textbook. And here, at least at the level that we are, if you are preparing for your UTME, this is a must that you have. College Biology by Idodo Ume. This book transformed the way I study biology. It sort of moved me from, studying biology, trying to memorize, trying to cram things, into, studying biology in a systematic way, such that I begin to understand, I begin to know. This is that biology. If you truly, desire to understand biology, most especially those going into the medical sciences, this is a textbook you should have. Idodo Ume, and it is not very expensive. You can just, check around at your local bookshops. You're going to have it. It's such a very, very, quality textbooks. It will transform the way you see biology, and you're going to move away from just trying to survive, trying to memorize biology. Because how many things do you want to memorize in your tiny in our tiny brain? Let me put it that way. You really cannot, because biology is an enormous subject. You have to study it from the standpoint of trying to understand, and not, trying to cram things up and memorize. So that said, this is animal cells. You can see when ribosomes are attached uh, to the endoplasmic reticulum, we call it rough endoplasmic reticulum. When ribosomes are not attached, we call it smooth endoplasmic reticulum. This is the mitochondrion, and this represents the crystal of the mitochondrion, which I'm going to talk. That represents the crystal of the mitochondrion. I'm going to talk about it at a close range. I'm going to talk, this is your Golgi apparatus, as discovered by Camillo Golgi. I'm going to talk about that as well, and a host of other things. But the thing is this is the nucleus, or the protoplasm of the cell, and every of the space outside of the nucleus is called cytoplasm. The cytoplasm is not an empty space. It is a semi fluid. So that ends that on animal cells. We will then, after drawing plant cell, we will take a tour looking at all of the cells inside out. So moving on, let's now quickly look at that's the animal cell. Let's look at the plant cell and how we go about things. Okay, this is the way I draw mine. Usually starts this way, and I go boom. Okay, now that I have that, I think I'm going to draw another. Okay, now that I have that, I'm going to try to draw, something on the outside. This here is at best how we, you know, how I can represent the plant cell. Firstly, you can see what this means. These are not just mere design. What this is telling us is that cells don't exist independently here. They exist in groups so that in numerous box-like structure as far as plant cell is related. So what this just means is that we have neighboring cell. There is going to be a cell here, 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 so that we, let's say we are talking about this cell here. So and we need to understand that yes, this cell has its cell membrane, which I need to quickly indicate, so that if I say this is the cell membrane, then this here is going to be the cell wall. This, if this here is the cell membrane, this here not touching on this line, this here is going to be the cell wall. And I'm going to show you pretty much later that the cell walls of neighboring cells are separated by what we call the medial lamella. But most importantly is that this is the, at least a basic structure for a plant cell. You can see the Golgi apparatus. You can see the large central vacuole surrounded by the membrane tonoplast. This is your tonoplast here. And this is not just an empty space. It contains cell sap, talking about water, mineral salts, sugars, and the like. That is what is contained in here. You have your nucleus, you have your rough endoplasmic reticulum, you have the ribosome. And worthy to note is the pit. The pit, you can see the pit. You can see the pit. So this ensures that materials are exchanged, between cells, connect and transport materials between adjacent cells. You're looking at the chloroplast here. Yes, a plant cell has chloroplast. Yes, I've indicated that. You're talking about the cytoplasm, and you see that because of the presence of the large central vacuole, most of the organelles of the plant cells presses against the cell membrane. So for short, this is what the basic plan of the plant cell looks like. So we're going to take a tour of all of these. Also worthy to, we will look at all of this at close range. Also worthy to note that talking about the plant and animal cells, they are not just, flat this way. No, they are more of solid shapes. For us to study them, like you can see displaying on your screen, for us to see the interior of the cells, we have to break the cell open. The plant and animal cells are actually solid shapes. They are not plain shapes as indicated on this flat marker board. As you see in in the right dimension, the plant and animal cells are solid shape. But what is of most importance now is for us to truly take a tour of these cells and look at the various little organs. That's talking about the organelles of the cell, describe them, look at what the structure of those various parts or organelles look like, and as well explore their functions. Yes, moving on. The next thing to deal with now is for us to take a tour of the eukaryotic cell, to look at that powerful machinery called cell, look at the various parts, look at how the various organelles, that's the little organs that we have on the inside, function, describe the structure, and ultimately look at how all of these organelles contribute towards the functionality of the cell. The first one we're going to look at is the cytoplasm. You need to understand that when you say the cytoplasm and the nucleus. The two together is what we call the protoplasm. It is not that you say the nucleus means protoplasm. No. The nucleus and the semi-fluid cytoplasm together, that's, you're talking about the whole internal content of the cell, is what we call the protoplasm. And you need to also understand that the cytoplasm is not an empty space. No, it is a semi-fluid which fills the interior of the cell. It is a fluid which fills the interior of the cell, excluding the nucleus. The chemical wealth of the cells, talking about the chemicals necessary for the functioning of the cell, is contained in the cytoplasm. You're talking about sugars, you're talking about amino acids, you're talking about ions. All of these chemicals are contained in the semi-fluid cytoplasm. The cytoplasm is not a vacuum. So what are the functions of the cytoplasm? The streaming. The semi-fluid in the cytoplasm is not just stagnant. It is streaming gradually. It is moving gradually. The streaming of the content of the cytoplasm brings about interchange of materials between the organelles that we have in the cytoplasm. So let's say certain chemicals are released. Okay, let's say in the case of the nucleus now, when RNA are being released and the RNA is going to the, let's say, ribosomes. How will the RNA, that's talking about ribonucleic acid, how will they move once being released from the nucleus through the nuclear pores of the nucleus? How will the RNA get to the destination? So this will be possible because of what we call the streaming content of the cytoplasm. So the semi-fluid that we have in the cytoplasm is not stagnant, is not fixated. It is constantly streaming. So the streaming of the content of the cytoplasm brings about interchange of materials between the organelles in the cytoplasm. And secondly, all the living substances, including the nucleus and all of the organelles, where are they suspended? They are suspended in the semi-fluid cytoplasm. In other words, the cytoplasm houses, accommodates the nucleus and all of the other organelles. So these are just about the two important functions of the cytoplasm. So let's move into the next one. We want to talk about the cell wall. When you talk about the cell wall, as you can see in the diagram on your screen, the cell wall is what separates a cell from another cell. That you can find this actually in plant cell, but the cell wall, like I wrote here, is the non-living outer boundary of the cell. It is made up of cellulose. Of course, that's a polysaccharide. It is absent in animal cell. And I'm coming back to this point, talking about the pores or the holes or the pits that the plant cell has, as you can see in the diagram we had in the lectures previously. Talking about the cell wall, it has tiny pores or holes or pits, through which nutrients pass from one cell to another. It just allows passage of nutrients from one cell to the other. Here, it is not that it regulates what can come into the cell and what can go out of the cell. It doesn't regulate. It doesn't control movement of substance in and out. What the cell wall does is via the pit, which contains plasmodesmata, which I'm going to talk about shortly after now. The cell wall allows free passage of material. It is absent in animal cell. And now, the cell walls of one plant cell and that of another plant cell are not in direct contact, as you can see on your screen. First, you're going to have a primary cell wall, which as the plant cell grows, a secondary cell wall will be laid down on the inside of the cell. Now, the two primary cell walls of neighboring cells are not in direct contact. They are separated by what we call the middle layer, which we call the middle lamella, which is made of another polysaccharide called pectin. So just take note of that, that the cell wall of plant cells are not in direct contact. they are separated by a polysaccharide called pectin. That's the middle lamella, which is made up of pectin. And when the rest of the cell are died, recall when we were talking about cell theory, looking at the work of Robert Hooke, we said that as old cells are being pushed out, they become filled with a waxy substance called suberin, which renders the cell impermeable to water, and ultimately the protoplasm of the cell dies, leaving only the cell wall alone. And I think that is the numerous box-like structures, tiny little rooms or small compartments, which Hooke had actually observed with his optical microscope, which he called or tagged cell. At that time, he didn't recognize the significance. You know, after being filled also with suberin, as those cells are being pushed out, they get converted into wood via a derivative present in the plant called lignin, in a process called lignification. So the cells that Hooke observed actually were only the cell wall. So when the rest of the cell had died, the cell wall remain, and that is why we say the cell wall is the non-living outer boundary of which the cell is made up of. And what are the functions of the cells? Of the cell wall? It allows free passage of materials between cells. It does not determine or regulate what may enter or leave the cell. And because the cell wall is made up of the polysaccharide cellulose, the cell wall of plants gives mechanical strength or rigidity, support and protection to the cell and then the plant as a whole. As you can see on your screen, look at those images very well. You can see where your plasma membrane is. You can see where your secondary cell wall is. You can see where your primary cell wall is. And you can see a line, an extrapolated line separating cell 1 from cell 2. Even look at this, an electron micrograph of the cell wall. You can see the layer labeled the middle lamella. So the two cell walls are not in direct contact. So moving on, I want to talk about plasmodesmata in relation to what we call symplast. Okay, moving on. When we talk about the plasmodesmata, singular plasmodesma, of course you recall that when I showed you the drawing for the plant cell, as you can as well see on your screen, you recall that you know, all around we have the pit containing plasmodesmata. Yes, plasmodesmata, that is plural. Singular means just a that's singular means plasmodesma. A single plasmodesma, you know, is just a passageway out from one cell to another. So when you talk about the plasmodesmata, it is embedded in the pit of the cell wall. You see, the cell walls possess minute pores, tiny pores, through which structures called plasmodesmata can pass. Now, what the plasmodesmata does is now that it allows materials to move from one cell to another, you know, cell. It allows materials to move from one cell to another cell, and you know, the process continues like that, sideways, down, up, and like that. So these connections are what we call living connections that exist between the protoplast of all of the plant cells, so that they are linked in a system called symplast. I'm going to take that again. When we talk about the plasmodesmata, that talks about the structures embedded in the pit of the plant cells. So since the plant cells do not exist like singly here, they exist in groups. So we talk of plasmodesmata. Now, what this structure does is that it now allows, as you can see in the function here, it allows free passage of materials between cells. So if it allows free passage, materials to pass freely from one cell to another, it now means that the protoplasm of this cell is connected with the protoplasm of this cell. And if we are to have a cell up here, the protoplasm of the cell will be linked with the protoplasm of this cell. So I said forming living connections between cells and allowing all protoplasts to be linked. That system is called the symplast. So that if I say the symplast of a plant cell, I'm basically talking about the linkage of the protoplast via the plasmodesmata, thereby allowing free passage of materials from one cell to another. So that ends cell wall. Where the cell wall is the non-living outer boundary of the cell, let's move to the living membrane of the cell. The cell wall is non-living. The cell membrane or the plasma membrane is living. It is a thin, flexible living layer. This statement alone, we said, is a thin, flexible living layer. Why are we saying the cell membrane is living? Okay, we had a cell like this. Okay, what you think is just a line. Let's say from here to here, what you think is a line is not just a line. If you look at the cell membrane under a very powerful electron microscope, what you're going to see is what we call a phospholipid bilayer. A phospholipid bilayer in the sense that this is more like phosphate, then at the tail here it's lipid, and then you have another phosphate here, so that it looks as though outside of the cell is phosphate, the inside is phosphate, and then in between the two phosphates is the lipid part. And of course, based on the trilaminar nature of the cell membrane, or what we call fluid mosaic model, this here are membrane proteins. Membrane proteins are what the cell membrane, looking at the design of the cell membrane, what the cell membrane does is that it decides what gets into the cell and what gets out of the cell. It decides what does not get into the cell, so that the integrity of the protoplasm is maintained with regards to what may enter into the cell and what cannot enter into the cell. These are things we are going to talk about when we look at cell and its environs, cell and its environs, or what we call movement or transport across the cell surface membrane. But the thing is this: when you talk the phosphate, did I say here the phosphate is the phosphate end is hydrophilic, or otherwise polar. The lipid part of the cell membrane is hydrophobic, non-polar. For things to get into the cell, this is like the barrier you have to cross before you can actually before things can actually get into the cell. But the thing is this: talking about the cell membrane is a thin, flexible, living layer that surrounds the entire cytoplasm and separates the content of the cell from their external environs and neighboring cells. The function of the cell membrane is to regulate movement of substances in and out of the cell. Take, for instance, glucose in the plasma of the blood needs to get into the cell. Oxygen needs to get into the cell. Carbon dioxide needs to move out of the cell. Amino acids of protein, AA, needs to get into the cell. Certain materials produced from the cell needs to get out of the cell. So constantly, materials are always moving into the cells, and the materials are always moving out of the cell. The cell membrane regulates what can enter and what cannot enter. If something is too massive, if it is too big, then it can't enter. Take, for instance, talking about glucose. Glucose is a much larger molecule. It's not going to be able to permeate the phospholipid region. It's going to be the membrane protein that moves it across. So the way as to how the cell membrane operates is that it regulates. It's a living layer. It's not a dead layer like the cell wall. It regulates movement of substances in and out of the cell. It protects the cytoplasm, and then it fixes or delimits Emphasis on the word delimit. It delimits the content of the cytoplasm so that even when the cell is in our body fluid, as it is being bathed by our body fluid, it's not as though, let's say, mitochondrion is washed off, let's say centriole is washed off. No, the cell membrane, cell surface membrane is there to delimit and fix the content of the cytoplasm. When we get into how substances moving and out of cell, we will talk more about the selectively permeable nature of the cell membrane to show you that, look, the cell membrane is living and it is therefore selective in how substances move in and out of the cell. But the important thing is that the cell membrane regulates movement of substances in and out of the cell wall, whereas allows free passage of materials. The next thing is endoplasmic reticulum, talking about compartmentalizing the cell, which is one of the basic distinguishing features between prokaryotes, that's talking about the bacteria, and eukaryote, talking about plant and animal cells. When you generally talk about the endoplasmic reticulum, it is what is responsible for most of the cell organization that we have. It is what is responsible for transport of materials within the cell and even out of the cell. It is responsible for even exchange of materials. It interconnects organelles within the cell. Looking at what is displayed on your screen, you can see a fine diagram, a micrograph representation of what the endoplasmic reticulum look like, talking about the rough endoplasmic reticulum when ribosomes are attached, and the smooth endoplasmic reticulum when little or no ribosomes are attached. Now, talking about the endoplasmic reticulum, recall that I said cell membranes, not membranes, cell membranes. Now, on the outside, you have the cell membrane. But when you move into this inside of the cell, most of the organelles are covered in membranes. The structure of both the cell surface membrane and the membranes of the organelles on the inside follow the same plan, phospholipid bilayer. So it's not really something we have to go into. But the important thing is the largest of the internal membranes, if you talk about all the organelles on the inside covered in membranes, the largest of the internal membrane is the endoplasmic reticulum, tagged ER. The interior of eukaryotic cell is packed with membranes, so thin that they are invisible. You cannot even see the membrane of the ER under the light microscope. You can only see that with the electron microscope. This endomembrane system fills the cell, dividing it into compartments. Look at the diagram on your screen very well. It divides it into channels, into compartments. So when we talk about compartmentalization of the cell, that is the work of the endoplasmic reticulum, compartmentalizing the cell, organizing it, making it possible to channel passage of molecules within the cell so that things are organized. Now, we all know that ribosomes, the function of ribosome is synthesis of protein. When ribosomes are attached to the endoplasmic reticulum, they are called rough ER. And at this point, you need to know that this particular endoplasmic reticulum is concerned with protein synthesis, so that proteins are produced and exported from the cell. So when you see ER to which ribosomes are attached, these are where proteins to be exported out of the cells are going to be manufactured. Then the smooth ER, not many ribosomes are attached. What this is concerned with is organizing internal activities, organizing molecules, lipids to be used within the cells, not out of the cells. Rough endoplasmic reticulum is concerned with that. But basically, the basic function of the cell, of the endoplasmic reticulum, is transport of products in the cytoplasm. It's just basically transport. Aside the fact that the endoplasmic reticulum compartmentalizes the cells, that divides the cells into compartments, creating channel for passage of molecule, the basic function of the ER is transport of metabolic product in the cytoplasm. Since proteins are attached to the ER, and we know that enzymes are made from protein, we'll say the endoplasmic reticulum help in the formation of enzymes and protein. Also, they interconnect the organelles of the cell. That is the interconnection center in the cell. You look at the spread. You look at the spread of, as you can see on your screen, you look at the spread of the endoplasmic reticulum and you realize that, look, when materials are manufactured, when molecules or chemicals are secreted from one side of the cell and needs to get to the other side, what we carry it is the endoplasmic reticulum. Thereby, we say it interconnects the organelles of the cell. Also, we say they provide surface for ribosomes to be located. Yes, talking about ribosomes. Ribosomes are the site of protein synthesis, and a single cell contains up to about 500,000 ribosomes to 1,000,000 ribosomes. Take, for instance, the hepatic cells of the liver, as is very rich in ribosomes, more than 1,000,000 ribosomes. When you talk about the ribosomes very rich in RNA, ribonucleic acid, very rich in RNA. And when you talk about ribosomes, they are actually made from the nucleolus, which is in the nucleus. That is where ribosomes are made. Like I wrote here, ribosomes are made up of special form of RNA called ribosomal RNA. The nucleus is very rich in RNA, and with that, it is able to manufacture ribosomal RNA, which the ribosomes are made up of. That said, when you talk about the ribosomes, although when we draw the diagram it's just minute like this, you need to understand that the ribosomes, as minute as it is made up of two subunits, a larger subunit and a smaller subunit, as you can see on your screen. This subunit join together to form a functional ribosome only when attached to a messenger RNA. Now, I need to talk quickly about DNA transcription there, but the important thing now is, as you can see on your screen, you can see the larger unit and the smaller subunit of the ribosomes. When attached together, form the ribosome. But the ribosome alone cannot function. Recall that the function of the ribosome is to make protein. The ribosome on its own cannot function, cannot make protein, except it receives instructions from the nucleus. Recall that the nucleus is the information center. It directs all of the things that happens in the cell. The ribosome on its own cannot function unless it receives instruction from the nucleus via the messenger RNA. When the messenger RNA is sent out from the nucleus and it gets to the ribosome, what the ribosome does is that it then decodes the information on the messenger RNA to know what type of protein to manufacture, to know the amino acid sequence to assemble to make the right kind of protein. Let me explain deeper. You see, DNA in the nucleus does not move out of the nucleus. There is DNA in the nucleus. There is ribonucleic acid. It doesn't pass out of the nucleus. Rather, when the nucleus wants to give information, what the DNA does is that it undergoes what we call DNA transcription. That is, that information, it copies it onto a special form of RNA called messenger RNA. So it is that messenger RNA that is being sent out of the nucleus. And when the messenger RNA gets to its destination, in this case, as the case of the ribosome, you see, DNA transcription is going to happen. The DNA transcription here means that a copy of the kind of amino acid sequence of the kind of protein that should be manufactured is copied as mRNA, messenger RNA, and that is now sent out of the nucleus. When it gets to the ribosome plus the messenger RNA, that is when the ribosome can now function to know the type of protein. Without the messenger RNA, without the instruction from the nucleus via DNA transcription, the ribosome is just about useless. It cannot manufacture any protein. So once the messenger RNA is available in the cytoplasm coming from the nucleus and is attached to the ribosome, then info on the messenger RNA is used to direct what sort of protein should be synthesized, and that is when protein synthesis occur. Now, both ribosomal subunit, both the large one and the small one, are manufactured in the nucleus. That is the reason why we say ribosomes are manufactured from the nucleus. Now, free ribosomes in the cytoplasm are to manufacture protein that will be used inside of the cell, within the cell, in the cytoplasm of the cell. But ribosomes attached to the endoplasmic reticulum, what they do is that they manufacture protein for export. Ribosomes are made up of special form of RNA called ribosomal RNA. You know, we have tRNA, that's transport RNA, we have messenger RNA, and we have ribosomal RNA. Ribosomal RNA makes up the bulk of our ribosomes, and the messenger RNA is just via the DNA transcription, where information is copied on a strand of RNA so that it is sent out, and that information is read at the right location in the cytoplasm so that they carry out the instructions of the nucleus. But basically, when you talk about ribosomes, it is minute, but then the function is straight to the point to manufacture protein. Recall I said DNA does not move out of the nucleus, but rather what it does is to copy its information, what it wants, on a special form of RNA called messenger RNA in a process called DNA transcription. So that ends that. We move straight to the powerhouse of the cell, talking about the mitochondrion, where the oxidative part of respiration actually takes place. Talking about the mitochondrion, that's in singular. In plural, mitochondria, usually, you have a good number of them in the cell, especially in very active cells like the cardiac muscles, like the hepatic cells that are constantly working. So you expect them to be very rich in mitochondrion. The mitochondrion, always referred to as the powerhouse of the cell, simply because it is in the mitochondrion that the chemical reactions of respiration takes place. Yes, that's the oxidative part of the chemical reactions of respiration. Glucose combining with oxygen to give carbon dioxide, water, and then adenosine triphosphates, simply energy. Now, this takes place in the mitochondrion, and that's why we refer to it as the powerhouse of the cell. As you can see on your diagram, the mitochondrion is actually sausage shape, and it is found in all eukaryotic cells, but you have more number in very active cells like the cardiac muscles. It is bounded by two membranes, as you can see, the outer membrane and of course the inner membrane, which is then folded into numerous contiguous that's rough layers called the cristae. Now, this reaction here will not be possible if enzymes are not present. Enzymes facilitate these reactions to happen in what is called citric acid cycle, or what we simply call Krebs cycle. Now, the enzymes that we need are, all on this folded layer called the cristae. That is why I wrote here, I said These reactions are accelerated by enzymes located. These reactions are accelerated by enzymes that are located on the crystal, as you can see on your screen. So what the crystal has done in this case, that imagine, okay, let's say this is the crystal, and let's say this is the crystal here. If enzymes are to be attached to this, where are we going to have more enzymes attached? Of course, the folded crystal here is definitely going to have more enzymes attached to it. So what this means is that the crystal have the effect of increasing the surface area for more enzymes to be attached. So the more the enzymes, the more the reaction is accelerated, and the more the energy that is produced here. So take a look at it very well. Chemical reactions of respiration, where energy is liberated, is going to take place in the mitochondrium, but that will not be possible if enzymes are not present. Now, in this case, enzymes are actually located on the crystal. So what is the function of the crystal? What effect does the crystal of the mitochondrion has? It is that it increases the surface area for attachment of enzymes. Also worthy to note here is the fact that mitochondrion contains DNA, and as such, when anything that contains DNA, what it simply means basically is it has the ability to replicate itself. But in replicating itself, the instructions for that is still going to come from the nucleus. In the case of a cell dividing, it doesn't mean when a cell divides, new mitochondrion will be formed. No. Before the cell divides, the nucleus will give instructions so that the mitochondrion doubles itself. The mitochondrion divides, doubling in number, so that by the time cytokinesis, the cell is going to divide, equal number of mitochondrion will be in the daughter cells. Take for instance, if a mitochondrion has 1,000 if a cell has 1,000 mitochondrion. Now the meaning of this is that when the cell is going to divide, the 1,000 will replicate itself, so that we now have 2,000 in the cell, so that by the time the cell divide, it will now be 1,000 to each daughter cell. But before the mitochondrion can replicate itself, the instruction will come from the nucleus. That is why it is not possible that you bring mitochondrion out of the cell and try to culture it and try to grow it so that it replicates. No, because it is still the nucleus that will give instruction for mitochondrion replication. But basically it is like this: organelles that contains DNA, like the nucleus, have the ability to replicate themselves. Organelles like the centriole contains DNA. Organelles like the chloroplast contains DNA. They therefore have the ability to replicate themselves. So that is that on mitochondrion. It is the powerhouse of the cell, because it is here that the chemical reactions of respiration takes place, so that cellular energy is liberated for the functionality of the cell. Moving on, next is lysosome. That's the enzyme storehouse. What the lysosome does in the cell is to store enzymes for various purposes: for digestion, for defense mechanisms, for breakdown of old organelles, for digestion of or recycling of molecules. As you can see on the board, when you talk about the lysosome, we call the lysosome the intracellular digestion center. Why is it a digestion center? It is because it contains hydrolytic enzymes. It contains lots of enzymes. It contains degrading enzymes. You see, one of the vesicles, that's enzyme storehouse, and it is formed by the Golgi apparatus. As you can see in the diagram displaying on your screen, in that diagram you will see the Golgi apparatus, how the lysosome is released from the Golgi apparatus, and as you can see, how it digests food vesicles. When the cell takes in uh, particles by phagocytosis, the lysosome is always on hand to digest those food particles. And of course, when foreign bodies like bacteria get into the cell, it is the function of the lysosome to move to attack such a foreign body, so it protects the entire cells from invading microorganisms. It is the digestion center. You can see in that diagram old or damaged organelle, like in this case mitochondrion. You can see the breakdown of that old organelle by the lysosome, just to mention a few. So when you talk about the lysosome, it is more of a vesicle formed from the Golgi apparatus, and it basically contains enzyme. It is an enzyme storehouse. The lysosome is membrane bound. There is a membrane that bounds the lysosome, because if it does not have membrane, the implication is that the hydrolytic degrading enzyme it contains will then be released, which is of course capable of digesting or breaking down the entire cell, for which reason we call the lysosome the suicide bag of the cell. You can see autolysis here, self-digestion of a cell by releasing content of the lysosome. If the lysosome were to lack membrane, and therefore it all the enzymes, the grading enzymes it contains, are then released, then it means it will lead to what we call cell apoptosis, that's cell death. It will lead to cell death, in which case we call this autolysis. Hence the lysosome is called the suicide bag of the cell. Okay, moving on. Lysosomes membrane bag contain high levels of degrading enzymes which catalyze the rapid breakdown of proteins. It is responsible for breakdown or digestion of protein, nucleic acids like RNA, that's ribonucleic acid, lipid, carbohydrate. It is all function of the lysosome to break down this. Lysosomes also break down old organelles, recycling their component molecules. The enzymes of the lysosomes have to be kept apart from the rest of the cells. It is membrane bound. If lysosomes release their content into the cytoplasm, their enzymes will destroy the entire cell. One of such cases in the tail of tadpole. A tadpole will have a tail, but as it grows, it metamorphoses into an adult frog, the tail will disappear. Why? Simply because the cells around that region have now released their degrading enzyme. Hence, the degeneration of the tail. Even the uterus as well, after childbirth, will return back to the normal size, all courtesy of the degrading enzymes of the lysosome. So what are the functions of the lysosome? Digestion. It is the digestion center in the cell. Autophagy. Unwanted structures, old and non-functional organelles are engulfed and digested by the lysosome. That is autophagy. Even foreign bodies like bacteria and other microorganisms, should they invade the cell, it is the function of the lysosome to use its enzyme to degrade them. Autolysis is self-digestion of a cell, which means the cell will release, the lysosome now will release its own content, its own enzymes, therefore causing the breakdown or the digestion of the cell. Hence, the lysosome is called the suicide bag of the cell. You can check the diagram once again, very clear. Okay, so basically the lysosome is the digestion center of the cell. Moving on to the next thing, the Golgi body or Golgi apparatus, from which the lysosome itself is manufactured or produced, and other secretory vesicles in the cell from where they are produced. So let's now move to Golgi apparatus or Golgi body as first seen by Camillo Golgi. The next thing is the delivery system of the cell, talking about Golgi bodies and when you have stacks of Golgi bodies, we then call it Golgi apparatus, discovered by Camillo Golgi, an Italian physicist, who first called attention to it. They are flat membrane bound sacs which are often interconnected with one another. They are especially found in cells that secrete, or rather, they are found in glandular cells. Cells that secrete materials are called glandular cells. Take, for instance, if this were to be the esophagus leading to the stomach, and then let us have a pancreatic duct like this. So that this be the pancreas. Okay, let the small intestine continue now. So that this be the pancreas. The pancreas is supposed to release insulin, right? So you need to understand that the cells here, because now they have to release insulin, we therefore call them glandular cells, and we expect them to be very rich in Golgi apparatus. Any cells whose function is to secrete or release chemicals or materials, those such cells are called glandular cells. And one of such is the pancreas. There are so many more in the body. You're talking about the various glands that we have in the body, and where all of the digestive enzymes are also to be secreted. These cells are very likely glandular cells, and hence they will be very rich in Golgi bodies. The function of the Golgi apparatus is in the collection, packaging, and distribution of molecules synthesized at one place in the cell and utilized at another location in the cell. As you can see in the diagram displaying on your screen, you can see the Golgi apparatus. It collects proteins, package it, and is now releasing it at the other end. You can see the work of the Golgi apparatus. It is to collect, package, and distribute. So basically for distribution, and I have said it, they are very much abundant in cells that secrete. That is where you have Golgi bodies. Take, for instance, if we talk about insectivorous plants, insectivorous plants, they attract insects. They are always usually very colorful. That's carnivorous plants. They are plants that grow in soils that are very poor in nitrogen, so they are brightly colored to attract insects, so that when they attract insects, they can digest the insect and use that to supplement their nitrogenous compound or protein requirements. How they do this is that the cells of those insectivorous plants are very rich in Golgi apparatus. As such, they secrete or release sticky slime, something that is more adhesive, like, so that once the insect lands on the bright colored insectivorous plant, on the sticky slime, it is then not able to escape till it dies there, and the plant digests that and use that to supplement its nitrogenous compound requirement. But the important thing here is that once a cell is a cell that secretes or produces chemicals, then you expect such cell to be very rich in Golgi apparatus, and they are therefore called glandular cells. So that ends that on Golgi bodies. The next thing is vacuoles, and then we will talk about plastids in plant, talking about chloroplast, leucoplast, chromoplast. Moving on to the vacuoles, it is found in plant cell, and it is the central storage compartment. It contains water, it contains sugar, it contains pigment, and it is osmotic in function. When I say osmotic in function, it has the ability to draw in water via osmosis so that it is stored in the central vacuole. It is surrounded by a membrane called tonoplast. As you can see on your screen, you can see the central vacuole of a plant cell. It is usually absent in animal cell. If found, what you basically have are food vacuoles, always very small, minute, and temporary. Okay, it is absent in animal cell. If any vacuole at all is found, it is small and it is more of a secretory vesicle which is temporal. It is not an empty space. Talking about the vacuole, it contains large amount of water and other materials such as sugar, ions, pigments, all this together forming the cell sap. So which means the contents of the central vacuole is called cell sap, and it is osmotic in function, whereby if it has a higher concentration than the environs of the plant cell, it has the ability to draw in water by osmosis. So we say the cell sap is osmotic in function, and the fact that it is osmotic in function, thereby storing water, it means it then increases the surface area of the plant cell as a whole, thereby pushing all of the organelles as against the cell wall and subsequently the cell membrane. Look at it. The central vacuole functions as a storage center for these important substances and also helps to increase the surface area to volume ratio of a plant cell. What that means is that if the central vacuole draws in water by osmosis, not if, when it draws it, when the central vacuole draws in water by osmosis, it then increases in size, pushing the other organelles as against the cell membrane and the cell wall, thereby increasing the surface area to volume ratio of the entire cell. The vacuole is surrounded by a membrane called tonoplast, but basically the function of the vacuole is to store nutrients, to store chemicals, to store materials. So talking about the chloroplast, it is the site of photosynthesis, whereby green plants manufacture or synthesize organic food by combination of simple inorganic raw materials. Take for instance, you recall this equation, photosynthetic equation, although the equation is a gross oversimplification because this is not actually what happened. It is more than this. It is more than this. Carbon dioxide combining with water to form glucose, whereas oxygen is given off as a byproduct. Now this reaction, whereby glucose or sugar is manufactured in the green part of the plant, takes place in the chloroplast. I said it is the site of photosynthesis containing the green pigment chlorophyll for photosynthesis. Chloroplasts are larger and more complex than the mitochondrion. Take note of that. But like the mitochondrion, as you can see, let's look at the image. You can see what it looks like. It contains thylakoid, a granum which makes up the thylakoid, and stroma which contains the enzymes needed for the reaction of photosynthesis. You can see that on your screen. Like the mitochondrion, the chloroplast contains DNA. So if it contains DNA, it then means that the chloroplast will be able to replicate itself. It will be able to divide and multiply itself. But even at that, the instruction and the control for when the chloroplast replicates itself, the instruction still comes from the nucleus. As such, the nucleus is always tagged the information center of the cell. But the thing is this: the site of photosynthesis is the chloroplast, and like the mitochondrion, the chloroplast contains DNA, hence the ability to replicate itself under instruction or direction from the nucleus. Other DNA containing organelles in plants are leucoplasts. leucoplasts are usually colorless, but then in some cases it stores starch. When we have excess glucose in the plant, excess glucose is stored as starch, and that is when you talk of amyloplast, for storage of starch. When you talk about the chloroplast, the leucoplast, and the chromoplast, they are collectively called plastids, and that is why I said plastids in plant cell, where the chloroplast is for photosynthesis, the leucoplast, otherwise called amyloplast, is for storage of starch, and chromoplast is responsible for the characteristic bright color of flowers and fruits. You look at it: chloroplast for photosynthesis, leucoplast for storage, and chromoplast, it contains yellow, red, because it is not all the pigments in the chloroplast that are actually green color. For example, if you see the color of tomatoes, what is responsible for that red color is actually the chromoplast. Moving on, okay, we've been able to talk about vacuous plastids in plant. Moving on is another organelle called centriole. The centriole as well contains DNA. So when we're talking about organelles in plant cell that contains DNA, you're looking at the nucleus itself, you're looking at the mitochondrion, you're looking at the chloroplast, you're looking at the centriole. These are organelles that contains DNA. That means they have the ability to replicate themselves, but the information for that replication, or the control generally, must still come from the nucleus. So let's quickly focus on the centrioles. Next on the list is centrioles, the microtubule assembly centers. When we say microtubules, as you can see on your screen, you can see your centriole at 90 degrees to each other. They help in cell division. They help form spindle fibers to which chromosomes are attached during cell division. And talking about centrioles, it is where microtubules are actually assembled. So that's why we say microtubules assembly centers. You see, each hollow tube is a microtubule, so it is then assembled together here to form your centriole. I say microtubules, long hollow cylinders of protein tubulin. So you see, just one hollow tube, according to that diagram, is a microtubule. So a microtubule triplet then, bounded together, forms your centriole. Each centriole is composed of about 9 triplets. You can see each centriole is composed of about 9 triplets. If you count it, 9 triplets of microtubules. So your microtubules makes up your centriole. Now, centrioles contains DNA. At the beginning of cell division, the centrioles replicate themselves. You can see, if an organelle contains DNA, the meaning is that it has the tendency to replicate itself under the information of the nucleus, under the directives of the nucleus. So centrioles contain DNA, and at the beginning of cell division, the centrioles replicate themselves, and two pairs migrate to opposite pole, one to this side, one to this side, to opposite poles of the spindle. When you talk about the spindles, the spindle is the structure on which chromosomes line up during cell division. Spindle itself is made up of what? Microtubule. Spindle is made up of microtubule organizing centers. So plant cells lack centriole. When you talk about microtubules, these are the protein tubulins, long hollow tubes. And talking about these microtubules, as you see on your screen, the interior of the cell, I want to quickly talk about what we call cytoskeleton. Let me quickly talk about cytoskeleton. When you look at the organelles of the cell, what keeps the organelle fixed, to spots? There are, just the same way we have skeleton in our body, you understand, to which muscles are attached in a cell. There is what we call cytoskeleton that crisscross the network of the cells, giving it shape and anchors organelles to fixed location, as you can see on your screen. That is to show you cytoskeleton. So that, imagine the nucleus, when we had to draw cell, the nucleus was at the center. So do you think the nucleus is just floating at the center? No, there are cytoskeleton holding it in that spot. The mitochondrion, there are cytoskeleton holding it to that spot. This cytoskeleton are also made up of microtubules. What are the functions of the centriole? Centrioles are important in cell division. They provide spindle to which chromosomes are attached. Centrioles help to assemble microtubules, which help to move the chromosomes during cell division. And also, when you talk about centrioles, they help to assemble microtubules. You can see microtubule assembly center. The microtubules are just long hollow tubules. Long hollow tubules. So what we then assemble them is the centriole. That's why we say microtubule assembly centers. Now, during cell division, they assemble microtubules that will help to move the chromosome during cell division. They also provide functional structure of cilia. Like when you talk in paramecium, centrioles provide functional structure of cilia, flagella in euglena, in chlamydomonas, in what we call a 9+2 structure arrangement. Let me see. Okay, I have that. You can see that on your screen as well. 9+2 structure arrangement. 9+2. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9+2 structure arrangement you can see on your screen. So what the centriole will do is that it will help to assemble this microtubules so that they form cilia, so that they form flagella in what we call 9+2 structure. This is very important. And so that ends that about centriole. It assembles microtubules for various structures, like the cilia, like the flagella, like for moving the chromosomes, and most importantly, for formation of spindle fibers during cell division. And it has the capacity to replicate itself under instructions or directives of the nucleus. So these are organelles that contains DNA. So that ends that. Okay, pit or plasmodesmata is the next thing, but we've talked about that. Moving on to the last and the most important organelle of the cell is going to be the nucleus, talking about the information center of the cell. And lastly, to the most important part of the cell is talking about the nucleus, the information center of the cell. It is the largest and most easily seen organelle within a eukaryotic cell. Talking about the nucleus, as you can see on your screen, you can see the diagram of the central nucleus. As you can see on your screen, you can see it. The diagram of the nucleus. It is the information center of the cell. You can see that from diagrams we had before now, that the nucleus via the nuclear envelope or nuclear membrane is continuous with the rest of the cell by joining with the endoplasmic reticulum. You can see the depressions on the nuclear membrane called nucleopores for materials to move in and out of the nucleus. But the thing is this: the nucleus is the repository of genetic information, and with that genetic information, it directs all the activities of the living cell. It is the nucleus. The nucleus simply contains genetic or hereditary information in form of DNA, and with that, They run and control all the activities of the cell. The red blood cells, once matured, has no nucleus. When you talk of something like paramecium, of course, they have mega and micro nucleus, whereas most cells have just about one or two nuclei. Um, Take note as well, the nucleus is surrounded by nuclear membrane, and that nuclear membrane is continuous with the rest of the cytoplasm via the endoplasmic reticulum. And the nucleus also contains chromatin. When you talk about chromatin, they are thread-like genetic materials which are not even visible under the light microscope. It has to be stained for us to be able to see them. When the cell is not dividing, it is more of a thread, and it's difficult for us to see. But when a cell is going to divide, you see the chromatin condenses visibly into chromosomes. Chromosomes are made up of genes, and genes are made up of DNA, which then codes for various activities in the cell. This here is a JAMB or UTME question. Okay, it contains chromatin and then one or more nucleoli, singular nucleolus. What the nucleolus does is this. I said it is where intensive synthesis of ribosomal RNA. Recall that I said we have various types of RNA. We have rRNA. This rRNA means ribosomal RNA, and this is manufactured in the nucleolus and sent out of the nucleus so that they make the ribosomes. So in other words, we do say ribosomes are made from the nucleolus. Then we also have messenger RNA. When you talk about materials that pass out of the nucleus via the nuclear pulse, you're talking about proteins. You're talking about messenger RNA. You're talking about ribosomes or ribosomal RNA. DNA does not pass out of the nucleus, but generally, when you talk about the nucleus, it is the information center of the cell, and it directs all the activities of the cell. So that is that on the nucleus. It is the repository of genetic information. Moving on, let's quickly look at the differences between plant and animal cells, as you can see on your screen. Talking about the differences. Moving on, let's quickly look at the differences between plant and animal cells. When you talk about a plant cell has a dead cellulose cell wall, hence definite shape. For animal cell, it is surrounded by a living membrane, hence it can change its shape. Whereas for a plant, it doesn't change its shape. Chloroplast is present in a plant cell. That is why we refer to them as autotrophs. Chloroplast is absent in animal cell. That is why we call them heterotrophs. Large permanent central vacuole present in plant cell. Vacuoles usually absent in animal cells, but if present, it is always small and temporal, more or less of a secretory vesicle. Food is stored as starch granules in plant cells in the leucoplast. Recall that we said the leucoplast is a plastic that is responsible for storage of starch. Food is stored as glycogen and fats in animal cells. Central absent in plant cell, central present in animal cell. Cytoplasm being the large surface area to volume ratio of the vacuole, the cytoplasm is pushed to the wall in plant cells. Cytoplasm spreads all over in animal cells. You have a large vacuole in plant cell, vacuole absent in animal cell. If present, it is usually small. And that is just about that, about the tour of eukaryotic cell. Quite a handful, you would say. And that ends that. So the next thing we're going to be looking at is the variety of cells, because the function that the cell will perform will be dependent on the type of organelle it contains. Take for instance, if a cell is so rich in Golgi apparatus, we call that a glandular cell, and that means it's going to be a secretory cells that is meant to release certain chemicals. So what I'm basically saying here is that the function that a cell is going to perform is dependent on the type of organelles that it contains, and for that reason we are going to be looking at variety of cells. Immediately after that, We'll be looking at forms in which cell exist. We'll be looking at organization of life because life exists in an organized form from the simple to the complex. And then we will now look at how substances move in and out of the cell. Remember, beneficial materials must move into the cells. When the cells secrete, right substances must be released from the cells. Waste materials must be released from the cells. So we are going to be looking at how molecules transport across the cell surface membrane, otherwise what you know as a cell and its environment, talking about diffusion, facilitated diffusion, osmosis, and the likes. From myself and the entire team here, it's bye for now.